20.
No, sorry, the one in Beaver.
Elizabeth, please conduct introductions for those guys.
All right. Hey, folks, we're getting started this evening. Thank you, Terry, for opening us up. If you couldn't hear the announcement, we are being recorded this evening, part of making this available for the public. And I'm going to invite folks to introduce themselves around the, we're going to go around the room first and then to our friends online.
Terry Song, District 3 Neighborhood, and also Chair.
Glenn Fee, Environment 1, and I am now as Vice Chair.
Rebecca Shaw, District 1. Elaine Stewart, Environment 2.
Mark Farrow, Builder, Developer, Representative 1.
Ashley Farrell, Business Rep 2.
George Marsh, Ag 1.
Alan Jesse, Ag 2.
Katie Ragsdale, Executive Assistant.
Joshua Bernier, Digital Solutions Tech.
Joey Newcomer, together.
Elizabeth Edwards, Chief of Staff at Water Services. I'm going to throw it to the back and
the Executive Management now is to the CEO.
I'm Ansel, Principal Engineer and Research Innovation.
Dale Fee, Chair of the Washington County Citizen Act Network.
Kathy Leader, Water Service Chief Financial Officer.
Joe Gall, Chief Utility Relations Officer for the district.
Shannon Huggins, Community Engagement and Water Services. Hannah Ferguson, Operations Specialist in Research and Innovation.
Whit Layton, Senior Program Manager and Research and Innovation.
Summer Sherman Berdinetti, Operations Analyst and Research and Innovation. Amanda McCary, Environmental Services Specialist with Water Services.
okay, to our friends online.
Ramesh Krishnamurthy, District 2 Rep. Apologies for no camera today. Sorry.
Stu Peterson, Business 2.
Sure.
Hi, Nisha George, Rep at Large.
And Dean.
Yeah, hi, this is Dean Brower. I'm an attorney over at Stoll Reeves.
Good to have you.
Okay, thanks, Elizabeth. Okay, first on the agenda is Scott Mansell and Blake Layton of the Water Engineering Technology Department. They'll be introducing the Research and Innovation Division of Clean Water Services. Okay, this is informational for the commission.
All right. It's all set up here.
Yeah, it should be.
Okay. Thanks very much for the invite to come present today. We're going to spend most of the time talking about eDNA and PFAS, but we thought beforehand we'd just kind of give you a brief overview of our R&I program at Clean Water Services, just briefly here. And so I will give a little bit about who we are, what we do, how we do it, and we'll give you a very high-level, tasting flight of some of our projects that we can, ask more about if you're interested before we dig into the eDNA and the PFAS. This is for your packet. We use a lot of acronyms, as you know, in our industry, so you have that for reference, but I'm not going to go through it here. So who we are, so we're a division within the Water and Engineering Technology Department. We're led by Peter Schauer, the division manager. He could not be here tonight, so we're pinch hitting for him. And all told, we're about 25 people, 25 researchers, and between us, we have a pretty wide range of expertise so that we're able to help contribute to a lot of problem solving for different departments, different areas. I'm not going to read through all these here, but you can see it's a pretty broad group. So it's a really fun place to do research. It's very applied, and so nobody's worried about getting tenure or publish or die like can be the case in a lot of research places. Everything we do here has a direct nexus to the benefit to Clean Water Services, the community, and the watershed. And because it's applied, we get a really, you get to see the benefit of your work rather than it being more abstract or more theoretical. And so that kind of ties back into our mission statement here, that our purpose is to serve Clean Water Services, the Tualatin Rivershed and community, And how we do that is by developing advanced methods, practical technologies, and data-driven solutions. And the ways that we serve them are primarily we do studies to help meet regulatory challenges, both current and coming down the pipe, to optimize our operations, do things more efficiently, more cost effectively, and to improve the quality of our services so we can help the environment and our ratepayers. And then we don't do this alone. We do this, I mentioned we have transformative partnerships. We go after creative and cost-effective solutions and impactful scientific communications. So we do present, we share our findings pretty broadly and pretty openly, as much as we can, trying to increase the impact and to bring more people along, have more stakeholders, work with other utilities, so that we, when we present to regulators or to any other stakeholders, we have a broader message. How we do the work is kind of multifaceted. So we have the research proposal program, which Blythe is going to talk more about in a second. That's basically anybody can propose a project to Clean Water Services and it will get reviewed and may get selected. Blythe will talk about that. We also are part of a lot of the capital improvement projects. So before something gets built, there's often a pilot study that needs to be done or a bunch of scenarios that need to be looked at or things that need to be optimized. And we get involved a lot on that. And almost nothing that we do in isolation. We're always partnering with other groups at Clean Water Services. So with our internal teams here, either they'll come to us, we'll come to them, we interact with a lot and they'll have a problem or a question or something that we want to study, something we want to solve, and we'll work together to solve it. We also do a lot of technology pilot tests to see what we want to bring in and what we don't, both in terms of treatment, sensing, things like that. And as I mentioned, we not just internally, we do a lot of partnering externally to help kind of broaden the range of expertise that we have, kind of share the load and share our clout, and in some cases, help bring in some external funding. So we do work with like the Water Research Foundation and groups like that. We do a lot of lab method development, which we'll talk some about today, and a lot of advanced data collection. So we kind of see ourselves as like the consulting firm that's internal to Clean Water Services. We work with all the different groups to help them solve their problems and work with them. And yeah, that's it. I'm going to pass it to Blythe.
Thanks. So Clean Water Services has an RFP or proposal program in which anyone in the district who has an idea that they want to execute can submit for funding. We have an annual process. For this, we have a rubric for the proposals. And some of the core requirements is, first and foremost, it has to solve or improve a problem here at the district. It has to be feasible. The proposal has to demonstrate that the author has thought it through enough that this can be executed. They're being thoughtful in their methods. It's a reasonable ask. And the proposals also need to demonstrate distinct benefits to Clean Water Services, whether it's in the realm of compliance or improving operations or demonstrating leadership, although that last one is not enough to get a proposal approved. It's always desirable, but really the most important thing is that first blue bullet up there, solving or improving a problem. That's what we're here to do. So I'm guessing y'all have heard about our roadmaps and probably seen some other ones. Okay, so these are the major elements in ours. I'm not going to read this, you have it in your packet. We're going to be going through some of these things in the next 6 slides at a really high level, just to, like Scott said, just to give you a little sampler of all the breadth of work that we do. So starting with our objectives around capital improvement, planning, and operation support, this is in the realm of our process engineering team. We have a tool that allows the engineers to estimate when they need to implement certain high dollar value capital projects. And that tool needs regular updating. And so the process team is really heavily involved in that. And they also work on projects such as piloting technologies that will allow us to increase the treatment capacity in the existing facilities without any capital improvement or expansion. I'm not gonna go through the rest of the bullets, we're just gonna hit that bullet once. And then over on the digestion and solids train side of the facilities, we have a few projects trying to understand the stability in the digester, developing a method to measure digester stability, and then also applying that method to evaluating different feedstocks to the digesters, whether it's fog or different food sources or what have you. And then in addition, on the solid side, there's other new technologies that you can read about. Check about that. All right, genetic monitoring. This is my piece of it. Those are folks in my lab up there in the photos. Looks like the very latest version of this slide. I did not get updated, but that's okay. Well, I had bolded things in here. I'm only going to tell you about microbial source tracking, which is when we're using DNA biomarkers from bacteria to identify the source of bacterial contamination, such as our E. coli exceedances at our MS4 sites. So we have a project working with our regulatory affairs department using that technology for that application. And then you're going to hear a lot about eDNA later today. On the next slide, I'm also going to highlight our molecular methods that we are using in a context of operational understanding of the treatment facilities. This is really novel work. We have Water Research Foundation grants supporting this project. And it's really important because the biological phosphorus removal process tends to go unstable for reasons we don't understand. And that process is really important to meeting our phosphorus permit. So having these new tools that will allow us to have a leading indicator of when these unstable events are coming and have a much higher resolution of data and information from these methods than we're able to get from our existing bench tests. All this is super valuable to our operators. And with that, I'm going to pass it to Scott.
I talk a little bit about these two, the research we do for regulatory compliance. So one is for evaluating ways to reduce our thermal load. Temperature is one of our pollutants of most concern right now. And we have a watershed-based solution where we rely on shade generation and flow releases to help offset some of that. And a few years ago, we did an analysis to optimize and come up with a strategy for how to best meet that. That was part of what went into deciding not to build the dam. It's been 5 years now. It needs to be updated. There's new regulatory requirements or things that have become a concern since then. There's new technologies, there's new ideas. And so we're starting a process now to update that. Similar is the update of the phosphorus TMDL and the aluminum standard. So Oregon has a relatively new aluminum standard that is going to make it difficult for us to use alum to treat our effluent to meet our very low phosphorus standards. And so, but at the same time, the phosphorus standard is based on a TMDL that's from 1988. And back then, the river looked more like a reservoir than it does today, had really long retention times, and it would have algae blooms. And it's a very different river today. And so for the last 4 or 5 years, we've been doing studies with DEQ to increase our phosphorus limit so that we don't have to use as much alum and observe, monitor very closely and model what's happening to the river. And as we predicted from the models beforehand, nothing happens to the river. It's not sensitive to small increases in phosphorus. And then part of other stuff my group does is advanced monitoring. So we do a lot of both gaseous and aqueous continuous sensing. And we, apply these to help solve problems. So we've deployed them in the collection system to help track down sources of pH spikes. We have them at rivers and creeks monitoring temperature and water level, soil moisture, groundwater levels, and things like that to help support our projects. And then we're always looking for ways to, either new sensors, new technology, open source things to help bring down costs. So that's an active area of research for us. I'm not going to get into our emerging contaminants because we're going to talk about PFAS a lot in a minute here. And then we do a lot of modeling studies. And so these could take the form of like water quality models of the river and Hag Lake that were used for things like the phosphorus TMDL update and the temperature modeling and kind of predicting what different scenarios will happen through climate change and population and if the dam weren't there and all that kind of stuff. We also do a lot of modeling of rivers and creeks for our natural systems group so they can quantify what the effect of various stream restorations will be, like what kind of difference it will have on stream shear and velocity and that kind of stuff, habitat. And finally, we have a means group that does a lot of conveyance system optimization. So one of the things they're working on is conveyance digital twin, which is a combination of real-time data collection and real-time modeling running. And so the combination of them makes kind of this, virtual conveyance system that we can ask questions of and test things and make predictions for when maintenance is going to be needed, when things need to be updated, and then be able to optimize capacity upgrades and stuff like that. They also have a conveyance equalization platform they're working on, which means that upstream of some of the pump stations in the dry season, there's capacity in the pipes. And so they're looking at ways they can make the pump stations run more efficiently by utilizing some of that capacity to increase the pressure on the pump to make it run more efficiently. That saves us time and energy, or saves us money and energy, I should say. With that, we can jump over to the PFAS and eDNA stuff unless there are any questions that people have or want to talk about any of the stuff we've done so far.
Any questions? Dean? Or Ramesh?
Yeah, so I have two questions, but let me ask you the first one. I might know the answer to this question, but I'm going to ask anyway. So how much of your work is local and specific to CWS needs, comparing it to something that's up level, something that's academic, or something that's generic research?
So 100% is local and focused on CWS's needs. We don't really do theoretical or that level of stuff here.
So all your funding comes from CWS budgets. Is that a correct assumption?
Not all of it, but almost all of it. We do get some outside grants. Well, I've mentioned the Water Research Foundation. We have several projects with. We've had a few EPA toxics, that kind of stuff.
Okay.
Mostly internal.
Yeah.
Which is another nice thing to have at a research institution, or compared to a more traditional research institution that would all be externally funded.
Yeah, I was just wondering, with the given current political environment, the funding is dependent on any external sources or external funding, and how that would affect some of the work that you guys are doing for CWS.
I don't think we have any grants from the federal government right now.
Yeah, I can speak to that.
Okay. Yeah.
We do currently have two grants. One is technically congressionally directed funding. We hope to put something actually before the board, the small summary, likely next week at the Tuesday board meeting. The other is, and that one was for about $1 million to address inflow infiltration issues. And then the other is a little over $500,000 related to a watershed navigator position with partners that would be over the course of 3 years. So compared to other partners, really de minimis. We're pretty fortunate in that space.
Okay. So if those fundings dry up, can CWS budget cover those things?
We have no indication that the funding for inflow and infiltration is at risk at this point in time, and that's a project where we need to actually stack a couple different sources in order to complete the full project. So that's in process. And then on the watershed navigator, we have some concerns because it's a FEMA grant. Mm-hmm. And we're waiting to see if the funding is going to come through on that one.
Yeah, thanks for all the good work, because I know this wouldn't be a good situation if we had to drop any of those projects because of some funding mishap. That was what was on my mind.
Quick clarifier on the RFP, I'm glad you addressed this. Where do most of those come from? So I think you mentioned, I'm paraphrasing here, I may have missed it, that it has to both benefit the watershed and CWS. So it seems like somebody would have to have a lot of knowledge of clean water services in order to submit something to that. But I might have been slightly off on that.
My understanding is that it's open to Clean Water Services employees, so staff.
Oh, I see. Okay, got it. I thought it was outside partners that might be applying. Okay, mm-hmm. Yeah, makes sense. Thank you. That makes sense.
Yeah, sorry if that wasn't clear.
Yeah, got it.
Any other questions in the room? Thank you. Segue into our PFAS efforts. Our next informational presentation is a discussion of PFAS concerns, regulatory developments, and how Clean Water Services is preparing for coming challenges through research, source control, regulatory advocacy, and outreach. Great. Elizabeth?
Yeah.
And Scott again, Amanda and Summer. Thank you.
Thank you, Chair Fong. So good evening, everyone. Again, Elizabeth Everett, Chief of Staff here at Clean Water Services. We'll each introduce ourselves when we get up to speak to our portion. But I have the great honor to kind of kick off this discussion because part of my portfolio is overseeing government affairs. And there's been a lot of action at the federal and state recently. So I'm going to cover the first couple slides and then turn it over to the real professional technical experts. This is an overview of what to expect. We've started adding a slide with all of the acronyms because it makes the subsequent slides less wordy. So hopefully this is helpful. All right, water PFAS, why do we care? I've been practicing saying per and polyfluorinated alkyl substances just for this evening. I really appreciate it. So also known as PFAS, also colloquially called forever chemicals. Well, it might feel like these are kind of a new phenomenon because they've been in the news a lot. We had movies like Dark Waters come out. They've actually been around since the 1940s. They're widely used, long-lasting chemicals, and the components break down very, very slowly over time. And in fact, there are thousands of compounds that sort of fall under this family of PFAS. They're found in water, air, and soil. And because of their widespread use and persistence in the environment, many PFAS can be found in the blood of people and animals all across the globe. And scientific studies have shown that exposure to PFAS in the environment can be harmful to human and animal health. So that is why we care, and a little bit about the fact that they're really everywhere at this point in time. So that caught policymakers' attention, right? It's a big deal. And the EPA, Environmental Protection Agency in particular, has been very active at the federal level. In 2021, they announced a PFAS strategic roadmap and have been putting forward draft rules and promulgating findings and rules since then. Last year, the EPA made recommendations for both aquatic life and drinking water standards. For PFOA and PFAS, EPA set a non-enforceable health-based goal of 0 in drinking water with an enforceable limit at 4 parts per trillion. I highlight that. Scott will get into a little bit of what this means on the scale and how we measure things, which is really quite challenging. Under public comment currently are those next 2, human health criteria for surface water and a biosolids risk assessment. These 2 in particular have the potential to be impactful for Clean Water Services. So on the human health criteria for surface water, EPA has proposed limits that are even lower than the drinking water standards, and in fact are over 1,000 times smaller than EPA's current ability to measure them, just to give a sense of how challenging it is to kind of capture and measure the prevalence. And so that's one of the challenges on that front. With the biosolids risk assessment, at a high level, the assessment found that the application of biosolids to farmlands pose no significant risk to the general public or general food supply, but they did find increased risk in a sort of hypothetical farm setting where individuals would only eat food grown on the farm, would only drink water out of a well on site, and that children on the farm would also consume some of the dirt or biosolids. So in that hypothetical setting, there were risks that they found. And those are both still under public comment. Layering onto the complexity of this, we also had an executive order that froze regulatory actions that hadn't been reviewed by the administration or designee. So we're caught up in that window too right now. There was actually a large announcement from EPA today with a lot of actions coming out of that agency. A cursory review, I didn't see anything pertaining to PFAS in that list, but this is breaking news today. Quick note on CERCLA. In 2024, EPA designated PFOA and PFAS as hazardous substances under CERCLA. Comprehensive Environmental Response, Compensation and Liability Act. While EPA's enforcement discretion memo made clear that the agency wouldn't be coming after or targeting clean water agencies for PFAS cleanups, I think there's a concern on the part of clean water agencies generally that CERCLA designation could potentially put clean water agencies at risk of third-party suit, despite being passive receivers of PFAS, right? We're not producing it, but we do receive it as part of our role in the overall ecosystem. So state legislative action. I'm not going to go into great detail on this because we had Tracy Rainey here with us a couple months ago. She spoke in more detail about some of these bills, but more just to highlight that State of Oregon is also working on addressing PFAS, specifically working on phasing out PFAS and products sold in state. And in 2023, we saw 2 bills pass, one related to cosmetics and personal care products, and the other around food containers, including polystyrene. All of these bills listed right now are still alive. It is before the first chamber deadline. And in fact, House Bill 3512, this Oregon Environmental Council Broad Product Phase-Out Bill, which includes everything from CWACs to artificial turf to menstrual products, kind of all together, that will have a public hearing tomorrow. So really active discussion and debate happening in the Oregon legislature, and we're monitoring this really closely. Final slide for me on this. Concerns about PFAS and policymaking. Our core mission, I think spoke to this in the R&I presentation, is to protect public health and the environment. And one of our values, at least I'm pointing to our Have a Best Day, we're guided by science. So we want to make sure that when lawmakers and regulators are looking at what are going to be the rules of the road here in Oregon, that they're making evidence-based, science-based policies and the right kinds of decisions for Oregonians. It is great that there's more attention about PFAS and the impact that they have on human health, the environment, animals. The concern is potentially having a pendulum swing and having overly restrictive rules that come out of reactions of events that happened in different parts of the country that have different contexts. So one of the things is that industrial sludge is not the same as municipal biosolids. If you're taking the waste product that comes out of a paper and pulp factory, it's going to be very different than what's coming out of the waste streams from domestic sources. And you'll hear later about a lot of the testing that we're doing to look at what is the impact of land-applied biosolids. In fact, we think there are a lot of benefits associated with it because it reduces waste, it reduces carbon emissions, it avoids the worst-case scenario of having to landfill these products, but we need to make sure that we're able to do that responsibly. And I think we're doing the science here at CWS to do that. And then part of my job and my team's job is to educate policymakers on what is within the realm of the possible, what is within the realm of appropriate policymaking that fits the context for Oregonians based on the industries that we've had here and the prevalence of the chemicals in state. So enough for me. I will pass it along to Scott. Yeah.
All right. So I wanted to get a little context here on some of the numbers and results we'll show. You know, Elizabeth mentioned these have been around since the 1940s, but have only gotten a lot of attention recently. Part of that is because they're really hard to measure. You know, when we're talking about part per trillion concentrations, I have a few things for scale on here that, you know, civilization hasn't been around for 1 trillion seconds yet. And so the laboratory methods and technology to be able to detect these at this kind of concentration, especially in things like biosolids and wastewater influent and really things that are hard to measure things in, there just aren't many labs that can do it. the country, and the technology is relatively new. But I want to go back to this kind of pathways of how PFAS gets to us from the environment. So PFAS are not naturally occurring. Every PFAS was made in a factory somewhere, or they're used in products in factories. That then are, get into products in our everyday lives, that can get into food packaging, stuff like that. Where Clean Water Services comes into this is, we receive some effluent from industries that can contain PFAS. We also receive leachate from the landfill that contains PFAS. But most of the PFAS we get is coming from what people are flushing down the toilet, washing off in showers and sinks and things like that. And so that's a really important piece that often gets missed. I added that arrow to this figure. All these versions of this tend to miss that is the main mass of PFAS coming to us. And so it's, PFAS that humans have already been exposed to that we're getting it. And so our concerns are what happens to it when we discharge surface water and when we land apply biosolids or recycled water. And so we've been working under this PFAS roadmap for the last several years, and it has these 5 objectives. One is to look at the treatment plants themselves and determine, what are the PFAS that are there, what kind of concentrations, where do they stand relative to relevant or regulatory concentrations, so we can know what our risk is. Tracking, are there treatment options that are being developed in the research, and understanding what's happening to them as they come to and from the plant. And then looking upstream. So where are they coming from? What can we do about it? And doing our best to address those. And then looking downstream. So when they are discharged in our effluent or our biosolids, what happens to them then? What is their fate, and what is the context surrounding that and the implications? And then, as I mentioned, we are preparing for coming regulations. We know it's coming down the horizon. As Elizabeth said, we want to make sure they're data-driven decisions. So providing data sets for regulators, we share our findings very widely. We get a lot of requests for data. We're building those partnerships through Aqua and with other utilities to really kind of help make sure we understand what's going on here in Oregon. And then underlying all of these, to make any of these possible for us to do, we needed to bring in the PFAS lab in-house. We had to do it ourselves instead of paying a lot of money to commercial labs to run the samples for us. So how we've been doing, since 2019, we've collected more than 1,300 samples, which is frankly an amazing data set. Much better or more than anybody else in the state by far, and one of the largest in the nation. And we get a lot of requests for that data. It's been very valuable to us in learning things, as well as those we've shared it with. We've successfully built the PFAS lab to have the PFAS method up and running in-house, thanks to Summer and the Water Quality Lab, that Summer we'll talk about later. And then we have a very active source control, thanks to Amanda and our source control group. We've seen measurable reductions in the PFAS that are coming to our treatment plants based on those efforts. And we have now, or will have, in the very near future, PFAS management plans in place for all of the industries that report to us. We've also done a lot of studies. I mentioned at the treatment plants, we've sampled industries, we've sampled commercial sectors, we've sampled domestic sectors to create mass balances of what's coming to us from where. We've gone hunting for sources of PFAS through the collection system and been able to find some major sources that way. We've sampled a lot in the rivers and creeks, including in urban and rural areas. And we've been studying soils and groundwater where we land apply our biosolids urea. So we'll talk about briefly of the results of all of these. And as I mentioned, we've been supporting Aqua and a lot of others' efforts. We provided training, data analysis for them, help with writing and advice to other utilities. It seems like at least once a month we have a call with someone calling asking for advice on PFAS from another utility. And so we're seen as a state and national leader on PFAS, and that's advantageous to us because we get invited to things that people talk about policy, where they talk about what the science behind things is. And we have some more clout when we talk to regulators and to the legislature. So diving into the data here. So objective 1, this is looking at the total of the measurable PFAS at our treatment plants. So the 4 colored ones are our 4 treatment plants. And compared to some recent data looking at the nationwide averages of some of these same list of compounds. So as you can see, Rock Creek has more of an industrial component. It's a little higher than the others, but compared to nationwide, even the black with the nationwide that reports no industrial impact, we're in pretty good shape here in Oregon. We do not have a lot of the problems that we found in a lot of places in the country where these things are manufactured. If we look at biosolids on the right, it's a similar story. We're order of magnitude below what the nationwide average is. A little comment on that though, on the difference between average and median. This is from a work that was done in Michigan, where they sampled all of the treatment plants, the biosolids from all the treatment plants in the state, and found that there's a handful here at the top that have much higher PFAS concentrations that are industrial impacted, whereas most of the domestic ones, the median of all of them is 13 micrograms per kilogram, which is exactly what we are in terms of our average. So in that way, we're a pretty normal looking domestic set of treatment plants. What Michigan did is they took an approach that in their regulations, if you can measure below 20 nanograms per gram or micrograms per kilogram of PFAS, then you can land apply without restrictions. And then if you're between 20 to 100, you have to land apply at a reduced rate, and you need to be doing source control to find where your elevated concentrations are coming from. That's the yellow bar here. And then if you're over 100, you can't land apply until you have found whatever your sources are and gotten down back into that green or yellow range. And with that, I'm going to pass it to Amanda.
Alrighty. Thank you, Scott. My name is Amanda McGarry. I work as an environmental services specialist with Clean Water Services. And one of the primary functions of our team is to implement the pretreatment program. And the pretreatment program aims to regulate the industrial users that discharge to our water resource recovery facilities. And as Scott's been mentioning, Clean Water Services has done a lot of work surrounding PFAS. And one of the tools, one of the strategies our source control pretreatment program is using is implementing these PFAS management plan requirements for all of our reporting industries. And really the importance of these plans is to characterize the wastewater. Clean Water Services needs to understand how PFAS is being impacted at the industrial users that we regulate. Another important component of these plans is we need to understand the reduction feasibility, right? That's not going to be the same for every facility. Product substitution may not be a one-to-one replacement, so we need to understand right off the bat what those strategies are going to be. And as I'm describing, the role of our program is to regulate, but it's also to teach. And with these plans, we kind of consider them a form of homework, right? We're having industries identify the sources. Where is PFAS coming from? Is it coming from chemical sources? Is it coming from structural sources? It also includes sampling requirements, and that's going to be a huge effort in having industries beginning to understand those sources. And I will say treatment is an aspect of one of the PFAS management plan requirements. We do have a landfill in our jurisdiction that sends us leachate, so that's wastewater that percolates through the landfill cells. But they also bring to us outside jurisdiction leachate. So it was through an agreement where we would continue to accept this outside jurisdiction leachate if they were to treat the PFAS mass that's coming to us from that outside boundary. And again, an outcome being pollution prevention. So the work that we do here is a lot of teaching and how do we prevent pollution into the environment. So here's a bit of that data shown here, the effects of our source control efforts. And we're looking at total PFAS mass. And we're looking here, starting first, FG, Forest Grove, and HB, Hillsborough. The PFAS mass is quite low, but I will say that's due to the sampling. Those are our two smallest facilities. So that's why there's a bit of that discrepancy. But if you look here at RC, Rock Creek, right there in the middle, we see a huge change over time of the PFAS mass coming to our water resource recovery facility, Rock Creek. And I want to start first with the gray bar, and that gray bar represents the semiconductor that we service in our jurisdiction. Now, this major semiconductor installed treatment, and notice how the PFAS mass coming to us from that source automatically dropped. It was a very dramatic decrease from the source. And interesting enough, that was not through Clean Water Services requiring treatment. This was actually through their own conservation efforts to implement water reuse systems. And again, here with the landfill being the pink bar here, another major contributor of PFAS to our jurisdiction. Notice how when the landfill leachate is shut off, how we dramatically see a PFAS reduction. And then again, we see the external leachate allowed, disallowed. And this is where we get into the data-driven regulatory decisions where Notice here about 50% of the PFAS mass is actually coming from the outside jurisdiction landfill, which is why we have now required as part of the PFAS management plan that fraction to be reduced by that amount. So they have that metric to meet for that reason. And we do anticipate the PFAS mass then to decrease from that sector with the installation of the foam fractionation unit, which is the treatment system they've elected. Now, on here for Durham, DM, this was an interesting occurrence here. So notice how in the first beginnings of the efforts of the PFAS program, we're seeing in 2019 and 2020, the majority of the PFAS mass was actually coming from the semiconductor and metal finishing campus. Now, the environmental services team and the research and innovation team collaborated to track PFOA through the sewer shed right to the source. And that was extremely groundbreaking to be able to track and trace PFOA. And ever since those conversations, these were ongoing meetings to identify with the industry where PFOA was coming from. We have yet to see Durham at that same level from that facility for PFOA. So again, showing the importance of ongoing source control, of proactive source control, and having these early discussions. And before I step away from this side, I do want to show and reiterate what Scott was saying, how once the largest industrial sources of PFAS are removed, the majority of PFAS is coming from residential sources, an exception to that Rock Creek facility, so the industrially impacted facility. And that really shows where are we going to focus our efforts, right? And that brings us to this slide. Coming up here, we'll be really working towards developing outreach materials. That is going to be huge for the environmental services team. And we're partnering with communication and community engagement here with Clean Water Services to develop sector-specific materials. So that's going to be for residential users, commercial users, as well as industrial users, to help them understand what PFAS is, where it's coming from, and try to get into that reduction. And in addition to that, and Summer will speak to this a bit more, we want to look at stormwater, right? We're really interested in seeing how stormwater is being affected by PFAS, specifically from industrial sources. So I have some highlighted here of the priority sampling initiative sites. And of course, that's going to come into additional outreach materials being developed. And I will pass it over to Summer.
Hello, everybody. My name is Summer Sherman-Bertinetti, Operations Analyst with Research and Innovation. Both Scott and Amanda kind of gave you a flavor of what we've been doing with Objective 1 and 2 of looking within the treatment plant as well as upstream, so our industrial sources that are coming to us. This is going to be kind of the first kind of section of talking downstream, where we're actually going to be looking at the rivers and tributaries. So we look a little bit upstream of where we discharge to the river, as well as the tributaries that enter the river at that point, as well as downstream from that. And so as you can see here, there are 3 plots that we have. I can use the pointer if I can get it to work. I have 3 plots, but basically what I want to show is that the bottom figure is the Tualatin River. And if you go left to right, it's upstream to downstream. You can notice that there's a lot of numbers everywhere. But what we have is we have these upper tributaries that enter the Tualatin River at those locations, as well as the lower tributaries that also enter the Tualatin River. And so I'll click through the slides, but what you'll notice is that with each click that I do, there will be another bar that kind of lines up next to it. We were able to sample the rivers and the tributaries in the wet season, so when it was raining, and the transition going from wet to dry, as well as the dry season, which is the summer season. So what we notice first off, this is just kind of the wet figure right here, is that in general, the upper tributaries have very little to no PFAS. While the lower tributaries, which are more urban versus the upper tributaries, which are more forested, have more noticeable PFAS in them. And if we look at the Tualatin River going from upstream to downstream, we also see that the Tualatin River starts very low with PFAS and gradually increases as we go downstream into more urbanized areas, also past kind of where effluent can come out as well. And so kind of showing the next few kind of clicks, we have the wet, transition, and dry. So when we notice, if we just look at the Tualatin River figure, which is down below, what we see is that in general, in the wet season, you have the lowest PFAS concentrations. In the dry season, you have the highest PFAS levels that we see. And so a lot of this has to do with dilution from the rain. And so PFAS levels actually It looks like it decreases because there's more water within the river at this point. While in the summer, it is very dry, and so the PFAS levels look like they increase just due to the lack of water there. So we wanted to look at, we do see an increase of PFAS, especially once we hit our effluent as well as our urbanized creeks. And so what is the dominant source of PFAS at that point in the river? And so we were able to do it in 2 different time frames. One of it is in the summer, which is what I'm showing right here on the left. And so what we see here is that the source of PFAS to the river in the summer in general is dominated a lot by the effluent. The Tualatin River is an effluent-dominated river in the summer, just based on the low flow. But then next up, we have Rock Creek, which is the urbanized creek that enters around the same point, as well as what the Tualatin River concentration is at that point as well. However, the wintertime has kind of a different story. There's more flow, there's more rain. These urbanized creeks, which can be very flashy, increase their flow and so enter the river with more volume. And so Rock Creek, the urbanized creek, actually becomes the dominant source of PFAS to the Tualatin River at this point. These urbanized creeks do not receive any influent or effluent. And so if we're thinking about limits to surface water, which Elizabeth mentioned earlier, we also have to think of what is actually available in the surface water. How does that impact us? Are there anything we can do just kind of around our watershed to prevent the runoff from going into the rivers? Are there locations that we can provide source control or educational efforts to lower this as well? On the right, what I'm showing here is PFAS concentration for the 2 PFAS, PFOS, and PFOA that most of the regulations are based over, as well as the EPA drinking water limit standard that Elizabeth also mentioned earlier. And so what I have highlighted here, I will kind of click through. In red, we can actually see basically these 2 regions, there's exceedances of both PFOS and PFOA at both of these, while both of these are actually our urbanized creeks. What I highlighted in blue is actually the Tualatin River after these urbanized creeks and our effluent are within the river. And so you can see that the urbanized creeks, which receive no influent effluent, are the only 2 to exceed these guidelines, while our effluent still is within the EPA range. So what we wanted to do is also kind of go from upstream to downstream of these urbanized creeks to see, is there a particular location in which PFAS levels increase? So we have our Rock Creek and our Fano Creek, which are our 2 most urbanized creeks in our watershed. And in Rock Creek, we go from upstream to downstream. You can see that there's a gradual increase of PFAS concentration. Rock Creek is in blue. We have our 2 purple kind of bars right here. These are 2 additional urbanized creeks that enter into Rock Creek. So it's Beaverton Creek and Dawson. You can see their levels of PFAS are a lot higher. And after they enter, PFAS concentrations in Rock Creek do go up. So we're kind of trying to track where this level of PFAS is increasing. And if we look at Fano Creek or other urbanized creeks, Fano Creek in general is an urbanized creek through and through. It starts very urban and ends very urban. And you can see that with its PFAS concentration levels. It starts at a higher level than Rock Creek and then kind of flat lines to kind of a stagnant PFAS concentration. And so with that, I will pass it to Scott.
Okay. All right, so if you were here last summer, Jared and I talked about our Commissary Research Site. You can see it on the map there. It's adjacent to the Tualatin River and the Durham treatment facility. We'll zoom in on it, but this is an area it was a former dairy that Clean Water Services bought years ago, and we've been working with partners to restore it back to the wetland. It still jurisdictionally is. But it's cut off from its flow of hydrology by all the development. And so we want to provide reuse to provide that flow back to the wetland. But there's no regulatory pathway to do that. So we've been working with DEQ to help develop that, to study this, to be able to show whether we can do this safely or not. And so we have a lot of research projects going on out here. And so if you were here last summer, you saw some us present on some of this. So I'm only going to talk about the PFAS work out there today. It's one of many projects, but we've been collecting PFAS data out there for 3 years of the groundwater and the soils. I'm not going to go into what some of the other ones are in this talk, but jumping right to the data. So in the brown here are the soil samples. You can see the control on the left that has never received any irrigation and is well outside and upstream of any of the irrigated sites. And site 1, 2, and 3 all receive irrigation and have different soil types. And so what you see in the soil is that, with the exception of the surface of site 2, which is highly variable for whatever reason, the irrigated sites are similar or lower than the control site. And looking at the groundwater, there's more variety in PFAS rather than just pretty much PFAS, which is all you see in the soil. But again, the irrigated sites are similar or maybe even lower than the control site. So this is not suggesting that the PFAS that we're finding in that groundwater has the effluent irrigation as a source. And that is in line with all the rest of the studies that we've been doing out there, with tracers and continuous sensors, microbial impacts and things like that. So that's been good news. We're going to do that for 1 more year. But what was interesting was all those PFAS we found in the groundwater. And so our question now is, okay, so where did they come from? Is that typical of the groundwater around here? Is there something special about that site there? And so we've been looking at this question, this urban signature. So this is looking at the blend of colors here is the different suite of PFAS compounds detected in the urbanized creeks that Summer just talked about, in Durham's effluent, and in these 4 wells. And you'll notice they're pretty similar, in both in terms of which species are there and their relative concentration for the most part. And so it's getting at, we have this kind of urban signature, we're calling it right now, that's in the groundwater, that's in these creeks, and even in the effluent, that we're trying to investigate what the source of that is. And so when Summer talked a little about some of our ongoing and upcoming stuff, that's an active area of research for us right now. All right, finally, we're going to talk about some of what we found out on the farms where we land apply our biosolids. So these are all privately owned farms. We have no control of the study variables on there. We sample with the permission of the landowners, but they've all been receiving biosolid application for 5 to 11 years, depending on the field. And we've sampled the same 3 depths. And every time we go out and sample, we also sample from the same field or a nearby field, a spot that has never received any biosolids as a control. And so most of them are in Eastern Oregon growing winter wheat, like the picture here. And then we have some growing mostly grass seed in the Willamette Valley. So jumping to the data here, focusing again on PFAS and PFOA, which are the main ones. You can see what we start with in the biosolids on the left here. And then we have Eastern Oregon and Willamette Valley controls versus the fields. So there is an increase in the soils that had biosolids applied for PFAS and to some extent PFOA as well. But we're very low. To put some context on the numbers here, Maine, the most conservative site versus or compared to or as far as what they're doing with their PFAS regulations, has soil screening levels for PFAS and PFOA of 5.2 for PFAS and 2.5 for PFOA. And we would always meet those for our PFAS and nearly always with PFOA. And so, we're down, really getting close to our detection limits with all these numbers for this. And to contrast that with what is often representative in the media of biosolids, like some of the dairies and things like that have been really contaminated. The big famous one is Stone Ridge in Maine, where the milk was contaminated and they had to close down and all that. Their soil concentrations they were seeing there were up to like 878 nanograms per gram. But that's often trotted out as representative biosolids, whereas that was one of the sites that had pulp and paper mill solids applied to it for years. And so this is kind of further confirming what we were seeing in the effluents, that we're in pretty good shape here in Oregon compared to a lot of other places. And with that, I'm going to hand it back to Summer.
Awesome. Scott. So we discussed this kind of last March, where we basically talked about building the PFAS lab, and that's really when we started to get up and rolling. But now, talking today, it is actually up and running. So building the PFAS lab, we put out a business case to actually bring this in-house. And so one of the great reasons that we were able to do this is we already have a water quality lab with amazing people that work there that have a lot of technical expertise in a lot of these things. The PFAS analysis, of which Scott mentioned earlier, is very expensive to run. And there are not very many places that can do it. And since now there's a boom of people wanting this data, these contract labs are getting swamped with all of these samples. And so turnaround times can be very long. We've even seen it up to 2-3 months for a turnaround time of a sample. And there can be poor quality assurance. A lot of the samples that we're looking at are industrial effluents, which can be a lot more difficult than a traditional sample, like surface water or drinking water. And so if we look at the figure that we have here, the return to investment estimated is what we kind of estimated for the business proposal, while actual is what we actually spent. And so if you recall from a figure way in the beginning, in just 2024 alone, we ran over 700 samples. And if we see our ROI by number of samples a year, we are really doing really well with it. And so it really made all the sense to kind of bring this in-house. We were able to answer questions faster. We're able to control our quality assurance. And this instrumentation is not only used for PFAS, it could be used for other emerging contaminants, such as 6-PBD-quinone, which is another big one, especially in the Pacific Northwest, as well as kind of other organics that we would like to look at. And so what I'm showing here, we did both a cross-lab validation as well as a validation with an external PT company, in which they basically send us an unknown. As you're running it, you do not know what the concentrations are. And you run it and report it. And they give you kind of what the concentration should be. And so in orange, we kind of have our acceptable limits for this PT. And if we see our blue bars, which is our actual data, is very close to that 100% recovery. This is a very hard analysis to do with very small detection limits. And so getting even closer than what a contract lab could report as an acceptable recovery was like an amazing accomplishment for this analysis. And so kind of Clean Water Services ongoing and next steps. There are a lot of projects that we want to work on, PFAS and stormwater, which Amanda brought up, that we will be starting hopefully this year, leading into next year, of domestic, commercial, industrial sources, looking actually more into outreach, creating this material, educational material for the public as well, source tracking in the sewer shed if we see a high concentration. Since we now have quicker turnaround times, we can hopefully find these intermittent sources a lot quicker than we could with a commercial lab. Expanding to other analytical capabilities, not only for other emerging contaminants, but there are other PFAS analytical capabilities that we could do in-house that will give us kind of a bigger picture to the concentrations that we are seeing. Sampling within the treatment processes, this will also help us optimize, if there's a place where the PFAS levels are higher, or even a place where treatment technologies might be most beneficial whenever they become available. Doing more urban signature studies, figuring out where is the source of PFAS coming from? Is it traditional for this region? This gives us a lot of information in which we could talk to regulators and being like, what is typical, what is atypical for our region? We also have a collaboration with OSU, in which we are working with them and their capabilities to pair with us for Thomas Dairy and kind of figuring out where is this PFAS coming from in the groundwater at our reuse test site, as well as kind of making these mesocosms, which are basically controlled experiments, boxes that we could control every aspect to answer questions of reuse, of using irrigation water, as well as biosolid applications. And it takes a huge team to do all of this. There are a lot of projects in PFAS, and so this probably doesn't even include every person that has touched any of these projects, but it takes a lot of people, and so we want to acknowledge a lot of their work. And with that, we will take any questions.
A couple kind of non-related or possibly related. I'm curious who you're getting all the samples from. Are they all from Washington County? You said the 500 to 600, I believe that was. And are you able to take those and use those for more collective knowledge for the region? And second question is, are you expecting a TMDL on this at some point?
For the TMDL, for the sample collections, most of the samples we collect ourselves. Actually, majority of the samples we do collect ourselves.
Okay.
So like Amanda and her team do go out to industries and sample at the industries at their effluent sources. So we have every single industry's particular one. For the tributaries, it's all within Washington counties, within our service district that we collect our samples from. Other ones like Thomas Dairy is within CWS's district, our treatment plants. We haven't run any external samples. It's all from our own collection systems.
So just to understand, you're showing the ROI on that. How is that ROI increased? Are you getting paid by who you're going to collect those from? Is that how the
it's avoiding having to pay the commercial labs $550 a sample.
It's simply from that. Okay,
Yes.
got it. So I misunderstood that.
It's paying for the cost of the instrument and the new staff.
I see. Okay. Thanks.
In terms of the TMDL, I'd be surprised. I don't know what it's going to look like. I mean, they are aquatic life standards and human health standards, so presumably there could be a listing which could lead to a TMDL, but there's thousands of these compounds. Yeah. And so you're going to make one for everyone individually, but treating them as a class, they have, over like 5 orders of magnitude of just the ones that have been studied of different toxicity. And so treating them as one thing isn't really accurate either. And so my guess is it's going to be like the mercury, where we have a TMDL, but everybody gets a variance because you can't possibly meet anyway. But I don't know. That's speculation.
Ramesh?
A quick question on the PFAS equipment that you showed in one of your files. Help me understand what's the kind of costs that are involved, costs of acquiring and setting it up, and also cost of ownership, like its usage and maintenance and stuff like that?
So the instrument itself was about $500,000.
It was like 400,000.
Yeah, and then with like the nitrogen generator and the solid phase extractor and all that. Maintenance and ongoing, it depends, because maintenance depends on how many samples you're running and all that kind of stuff. And for these first few years, we have the maintenance contract built into the price that we paid. And so we're getting free maintenance on it for now. But we've estimated, I can't remember the amount of man hours or person hours it would take to do these versus the samples. That's how we came up with our return on investment. And it was somewhere in the neighborhood of like $150 a sample instead of the $550, something like that.
Yeah, no, thanks. That helps understand a lot. So this is an equipment that the capabilities are not available now, or I'm trying to understand what's the need for this new equipment? We don't have anything at all, or we have something, but we need something better?
At Clean Water Services, like why we needed this equipment at Clean Water Services versus using something we already have.
Okay. Thanks a lot.
I think it was a clarifying question.
Yes, that was a clarifying question. Yes.
We didn't have a tool
Yes.
in our lab that made these measurements. So
Yes.
we were relying entirely on commercial labs, which I forget who referenced it, but they were made very backed up.
Yes.
And each sample individually was expensive, plus the shipping.
Yeah.
To make sure.
Overnighting a cooler of ice. Yeah.
Yeah, and even when we would overnight a cooler of ice, like with the PFAS bottles, a lot of time, there were a couple times where the shipping company ended up either not being able to deliver or losing the package or holding it, which then you have to decide whether running those samples is even worth the $550. But people already spent a lot of time and effort to collect those samples. So there were a lot of mishaps like that.
We ended up throwing out a lot of data, unfortunately. Could we do our own internal QA of it? And we don't want to use data that, doesn't pass muster for us. And when you're paying $550 a sample and have to throw out the data, that's really frustrating.
No, I agree. More than all of them, having the facility available and the quick turnaround time. sometimes time is money. The time itself is worth it. That's what I was looking for. Thanks.
Still on mute.
Sorry, did you say Nisha?
Yes.
Okay, great. I had a question about when you look at the sources of the PFAS, what happens if there is a source that I guess there isn't like other regulations saying, you can't have this type of discharge, like the rules around that haven't been put in place yet. So this, whatever industry or particular product can be contributing to the PFAS, but then the source. How do you stop additional contributions from that, industry or product or whatever? Does that make sense?
Yeah, that does. No, that's a really great question, and it is a challenge. And that's exactly why we have these PFAS management plans to start the conversations, right? Since there is no regulatory requirement at this time to minimize or to reduce or to treat PFAS in wastewater. So everything we're doing is from a voluntary standpoint, a voluntary effort. Though an industry is required to characterize the wastewater and to follow the conditions of their permit for monitoring for PFAS, you're completely correct. There is no regulatory requirement for them to reduce PFAS. So really we're relying on our partnerships, we're relying on our ability to implement effective outreach, and that's where we stand right now.
The regulations are coming. So part of it is helping them also get ahead of the regulations so they can make smart decisions instead of being, forced into something later.
Okay, thank you.
My question is also about source control and management. I be correct, but I did not know that there was treatment available for PFAS in water. So how are these management plans actually dealing with PFAS when it is above these limits?
So I will say for the one facility, so only one industrial user is implementing the treatment technology. I will say that treatment technology is actually a removal technology. It isn't a complete destruction technology. Now, that's not to say there isn't a destruction technology available, and I'll let Scott chime in on that. I think you know a bit more.
There are at the kind of pilot emerging phase some technologies that are really expensive and really energy intensive for destruction. You basically have to get the temperature really, really high and often the pressure really, really high too. But for like the concentrated stream, like the one Amanda is talking about, the foam fractionation is just a separation. And so, these are surfactants, so they come up with the foam, you blow bubbles to the water, and you skim off the top, and you can remove a sizable portion of the PFAS. And it only really works if there's a pretty high concentration of PFAS already. And then like for drinking water, you can, there are removal technologies like granular activated carbon, reverse osmosis, and some of these other real expensive things. They'll remove PFAS. You still have to deal with, what you do with the used carbon or the brine from your membrane. But for wastewater, there's nothing really out there yet. And so, yeah.
You're right. Because I guess where I'm going with this is like if we have this, removed material, which is then presumably having to be deposited elsewhere and eventually leachate back into our water stream. So is this just like a cyclical scenario that we're into?
Yeah, and that's kind of the fear, right, of making sure that doesn't happen. In this case, that highly concentrated PFAS volume actually gets transferred to a high containment hazardous waste landfill. So similarly to how they manage other hazardous waste, that's so that it doesn't impact the streams.
Okay, so next chapter. I was lucky enough to hear a speaker a week ago who does work on endophytic bacteria, bacteria that live within cells inside plants. They're not in the soil and in the roots. They're in the plants themselves. She might be a really interesting research partner. Her name is Sharon Doty, and she's at University of Washington. I just sent something to Shannon a few minutes ago, and she probably doesn't even know it yet. But I downloaded a paper while you guys were talking tonight, and there is some early work happening now with fungi and bacteria and plants actually breaking down the PFAS. And if Jared wants to submit one of your RFPs, that might be some really interesting work to do in the Thomas area and see if you could begin literally busting it up and making it go away.
Yeah, I think I see in the literature there's stuff like that that's coming up. And so I don't feel like we're too many years away from some form of treatment, but it's nothing scalable yet. And so, yeah, and then when it is, what the costs and energy requirements and all that will be, I don't know. But
I certainly hope the right plants are going to be a lot cheaper than osmosis.
Oh, I would hope so.
Any other questions? Great, thank you.
Thank you.
Our final presentation is an introduction of environmental DNA, also known as eDNA, a description of how Clean Water Services is using eDNA to meet regulatory and partner needs through 3 case studies. Blythe and Hannah.
Hello, everyone. Okay, you can see me over there. So I'm starting it off. I introduced myself before, but I'll do it again. I'm Hannah Ferguson. I'm an operations specialist on the research team, and I work in our molecular lab, of which Blythe runs. So we're going to be talking about environmental DNA. In particular, we have 3 kind of use cases or projects that we've been working on to share with you. Make the slides progress. Okay, so our agenda. We're going to give a brief introduction to what environmental DNA. Raise of hands, who has heard of eDNA? Cool. Most of the room. Exciting. Okay. And then we're going to talk about 3 case studies that we have. Coho, which is obviously a salmon. Emerald ash borer, which I think you guys recently had a presentation on. Cool. So you guys are very aware of it. And then finally, Blythe will finish off with metabarcoding, which is a slightly different eDNA technology, and how this is planning to be implemented in our Clean Water Services and BDS permit. So in your packets, there are a bunch of abbreviations. Like others, I'm not going to go over these, except for one. I'm going to highlight PCR, which is polymerase chain reaction, and that is a technology through which we take DNA and amplify it to an amount that we can actually identify in a sample. And so that's what all of this, all these studies rely on, is this basic technology in molecular biology. So what is eDNA? Pretty simply, it's genetic material that's left behind in a habitat. So in our little schematic over on the screen, we've got our nice stream, we've got like a deer or an elk, I'm not sure what that is, some kind of ungulate, and some other species that are hanging around. And all of the double helixes you see are DNA. We can think of them as breadcrumbs. And so as organisms move throughout their environment, they leave pieces of themselves, whether that's feces or tissue that sheds off. Gametes are a common high level of DNA that gets left. And so within that environment, we can go in and take a water or soil and sediment, air, even spider webs. We can take these kinds of samples and identify DNA that is found left in them. And so what this allows us to do is basically identify species that are in an environment without physically setting eyes on them. And this becomes incredibly powerful when we have species that are at low abundance. So things like invasive species or endangered species, it can be really helpful. It also, on the flip side, is helpful in that it allows us to characterize entire biological communities. And so we're not going to get into much the different types of eDNA and how we actually analyze it. But these tend to be kind of 2 different ways that we take a molecule and figure out what it is. And this is more what Blythe will be talking about at the very end. So why use eDNA? It's simpler often, it is cheaper, and it's faster than traditional monitoring methods. There are times that it is complementary, and there are times that it is better than some of the traditional methods. There are things that it can tell us, things that it can't necessarily tell us, such as abundance. But one of the really great things about eDNA is that it allows us to take more samples, and often we can take samples in areas that we don't necessarily have access to private land. So when we're thinking about something like spawning surveys for salmon, those surveys, you have to have landowner access. Whereas if you're taking an eDNA sample, you can take that from where the road crosses, so where there's right-of-way access to a stream. Okay, so as we're talking about, I've kind of alluded to this, but both coho and emerald ash borer, we're using a single species approach. This is very similar to like COVID sewer surveillance tracking that I'm sure many of you are familiar with. And meta-barcoding, this is a meta-barcoding approach, so it's a many species approach. So our coho eDNA study. So this was really motivated by DEQ's concerns about the outfalls, our outfalls from our water resource and recovery facilities as potential thermal barriers to coho migration. In particular, they were worried about migration as a whole, but the time period of when there was concerns really about the thermal migration or the thermal barrier was during coho migration season. And so we developed an in-house PCR assay in our molecular biology lab that we could use to identify the level of DNA, of coho DNA left in water samples. And we designed a 2-year study where in the first year, we wanted to look at and get a time series of when they were downstream of one of our outfalls. So for this first kind of 2-year portion, we focused on the Rock Creek outfall. And really why we needed this first year to understand when they're there is that the best information that we had, other than just general observations that folks had had in the upper watershed, was when coho are coming over Willamette Falls. So that's when they're in the Willamette River, but we didn't have information about when they enter the Tualatin River, and better yet, when they have moved, almost halfway up the Tualatin River. Does that happen in a day? Does that happen in a week? Does that happen immediately after they come over the falls? So these are the kinds of questions that we didn't have answers to, that we're starting to have, as I'll show you in a slide or two, some answers to, and that could directly impact our ability to kind of manage river temperatures and how we discharge in the coming future as climate change increases the temperature of the river, making it hard for species such as salmon that are cold water species. Anyway, so in our second year, we took higher frequency sampling above and below the river. For this presentation, I'm not showing you the above and below comparison. We don't have that quite finalized yet in terms of analysis, but I am showing you a 2-year time series of when we see coho DNA. Okay, so now we're going to walk through the time series. On our graph, we'll have a dark blue color, and that is our DNA quantity. So that's what we physically measured within water samples that we took at a couple of locations on the Tualatin River. In green, we're going to have the daily number of coho that are coming over Willamette Falls. So those are physical fish that ODF and W have counted. And then we have time on our X. So we start in July of 2023 when we initiated this project and have it running till, I think, February is our latest sample that we've run. So when we look at the first year of data, this is what we find. So in kind of the late summer and then in the spring, we find pretty low concentrations of DNA. And then we have these two we have a peak and then another peak. And we've identified this first one. We believe this is our migration peak of fish coming through. And we believe this second signal is spawning. And we believe that because of the timing. It would be really late for coho to be running. And there are studies that are done outside of the Tualatin Basin, as I'm sure many know, that look at coho spawning and the timing of that. And that second peak really aligns more with those studies in other areas in Oregon. If we look then at how this compares to the fish counts at Willamette Falls, I've now popped in that green data, and we have about a 1-week delay between when the fish come over the falls to when we start to see the DNA come up in the Tualatin. And then the last thing that we wanted to look at is kind of some of those environmental cues, and in particular temperature, and wondering how it might affect migration timing. There's a really great study that was done in the North Umpqua River on their coho population that found that temperature was the main driver to initiate migration. And so that was one that we wanted to cue in on. And in particular, they identified for that Southern Oregon population that 19 degrees C was a temperature at which it initiated. And so when we look at the time where the mouth of the Tualatin drops below 19 degrees, we see that most of our DNA concentration is coming after that point. So this starts to point to maybe some environmental cueing that we can pay attention to. So then if we look at how these trends compare year to year, this is our second year of data. And so we find, again, fairly low concentrations in those shoulder seasons, though we haven't really gotten to the spring of the second season. But we see that 1 week delay again between the fish counts over the falls and what we see in terms of our DNA concentration. We also have the 2 separate peaks of migration and spawning. And then if we look at temperature, we see a similar trend where most of the DNA is coming after that 19 degrees C. So in terms of the impact and where we're going moving forward on this is that what we're finding is that this indicates some environmental triggers for coho migration. We only have 2 years of data, but we see similar trends. And so we know and can imagine that this will help us manage the thermal regime in the Tualatin River. And this helps us protect salmonids under climate change, as I was kind of alluding to before. So our next steps, we're going to monitor the migration past Durham, so that our lower down treatment plant this year, starting in the fall. And we actually are also going to take a few samples closer to the mouth of the Tualatin to try to understand when they are actually coming into the Tualatin, since that's going to initiate the run up the river. And then we also want to keep in mind how patterns are shifting in response to changes in water temperature, right? So we know that the water years change year to year, and this is going to affect both our ability to meet a thermal TMDL, but also to manage for coho. So with that, I'm going to pass it to Blake.
All right, so now for something completely different. I'm going to give you a different vignette of how we're applying eDNA to solve important questions and problems that we have in our watershed. I'm really glad you guys already had a primer on the emerald ash borer. I don't have to convince you what a big problem this is. Clean Water Services, one bullet point of background. It is a pest potential to really decimate our ash trees. It was first detected in Forest Grove in 2022. And currently we have a joint project going between Clean Water Services, the Oregon Department of Agriculture, and the Oregon Department of Forestry to use our PCR techniques in our lab to help monitor trees for the presence of emerald ash borer. And the goal is to be able to use eDNA to determine whether trees are infected or not, because it's kind of hard to tell. Actually, you have to kill them. You have to cut down the tree and look. And so the methods that we're developing are allow us to sample a tree without cutting it down. So here are some results. So this was a study that we did where, 18 trees were sampled. And on the left in the columns, that's the result of debarking, whether they observed the presence of emerald ash borer or not on those ash trees. And then on the right-hand side of the columns, you'll see the eDNA results. And we had 2 trees in which we had both positives with both methods, and then a number of other trees that were both negative. Only 1 tree was positive for EAB by debarking and negative by eDNA. And a whole lot of trees were positive by eDNA, but negative by debarking. And so we're doing some additional work this summer to try to discern if there's any concern about deposition or DNA left behind by bugs that are no longer currently present. So we have additional experiments going on this summer, but we were really excited and encouraged by these results. First of all, it works. The method works. It's a roller method where we take a paint roller and disinfect it, bleach it, and roll it around the tree at whatever height we expect the beetles to be at, and then put that roller in some buffer and massage it all around, get all the material off of it, and then treat that buffer like a water sample, filter it, and then process it the way we normally do for our eDNA water samples. And it worked. So our next steps, and there's a photo of Matt from ODF doing that method I just described. So we're going to continue this project this summer, as I mentioned. And then internally, we're also coordinating with our partners in our Natural Systems and Environmental Stewardship Department, NSES, and doing a study on trees here in our watershed. And then we hope to potentially be able to offer this to our outside collaborators, other partners in the region. We don't know of any folks who really are doing this. So, we want to be able to help our partners because, of course, more regional coordination on this is going to help us get this species under control. Okay, so now shifting gears again, talking about metabarcoding. So just to reiterate, this is a different technology now. Historically, we've used macroinvertebrates to meet our biological monitoring requirements for NPDES permit. These guys are small aquatic animals and aquatic larval stages of insects. They're the little critters at the bottom of the stream, crawling between your toes when you go wading. These guys are good indicators of water quality because they have varied sensitivity to different stressors, particularly pollution or dissolved oxygen. They're also good indicators because they can't move. They can't outrun these stressors. They're kind of stuck in their environment. So historically, we've used these macro surveys, and it's a non-trivial effort to collect them. And then we calculate these different metrics. And the result of this is we get some kind of estimate of the health of that stream. Well, the problem is that these metrics were designed for steeper, higher gradient gravel bottom streams. Well, most of the streams in our watershed are flat and silty. And so we get not informative scores on these macro metrics every single permit cycle. So we were in search of something better. In the last 20 years of surveys and the last 4 permit cycles, we've had no real change in any of this information. And yet we have been doing a lot of work and restoration work in the watershed. So those efforts are not being characterized by our current metrics. So, and for eDNA and metabarcoding, this is going to give us a much greater abundance of information. With this method, we can get information about many different groups of organisms. From a single sample, we run PCR for these 6 different groups. So we're profiling across the entire tree of life. And this is not a presence absence, is this species here or not, like we were talking about before with the coho and emerald ash borer. This is sequencing these PCR products. And so it's like a barcode, and you can tell which species that piece of DNA came from. And so then you get a profile of the biodiversity in that entire ecosystem across all of these, levels of life. And this is an example of what the results can look like when you get these taxonomic profiles. Again, the entire tree of life, but here in a circular format. And so the goal with this is we're going to build a metric that's based on much more diverse groups of organisms, not just the macroinvertebrates stuck at the bottom of the stream, but everybody who's using that habitat, including groups that we're more likely to see in those silty, slow, flat rivers and lower tributaries. So where we're going with this in the future, this is really exciting. DEQ has already agreed to allow us to use eDNA data for biological monitoring to meet our permit requirements. So for this next permit submission, we're doing both the macro surveys we've always done and also eDNA as well. And we've already collected those samples in parallel with the macro samples, and we'll be analyzing those for this. And so in preparation for that, we're developing a metric based on eDNA, which will allow us to track these changes in ecological health. I already mentioned the paired samples collected last fall. And so the next steps are to get those samples analyzed, get that sequence data back, and then do the really challenging work of building this metric. So it's going to be based on indicator species. There's other what's the word I'm looking for? Archetypes we can look to, other jurisdictions that are using eDNA as a health metric for different water bodies. So, we don't have to completely start from scratch here, but we do want to build something that's specific to the Tualatin Basin with all the species that are present here. And with that, we will take any of your questions. I'm sure you have a lot. Yes.
I'm just curious, as I think USGS would be really interested in what you're doing here. Are you partnering with them?
We are. In fact, we have been working closely with the Powell River Watershed Council to develop a eDNA cooperative group across the entire Upper Willamette Basin. So we have in January, we held a workshop, and how many folks attended?
About 40.
About 40 folks attended from, it was federal, state, local public agencies, NGO, watershed councils, utility, just really a wide swath of practitioners who are all using eDNA for conservation or invasive detection or, all these different applications. And so the goals of coming together is sharing data, sharing resources so we don't have to replicate efforts in certain regions, right? When we have multiple stakeholders and around a particular watershed or whatnot. Data sharing methods, standardization, so that those data are more comparable across studies. We have a lot of goals, and we're continuing that effort with a webinar series and planned annual workshops. So yes, we are in communication with our local regional partners on this topic.
It's amazing to think that eDNA can provide such rich information across like all those taxonomic groups and characterizing the health and the diversity and community structure in an area. It's just wonderful.
Really fun to see that. And it's kind of a game changer, I think. Curious about what the indicator species are. Are you developing that list? Or is there a list that you're basing it on?
It's a great question. We have from our pilot a list of some indicator species that were put together by John Getz, the third, who works at Clean Water Services. And he went out to a number of partner organizations and asked them what kind of native species they thought would be important. So there's a number of mussels on there. There are some macroinvertebrates, some of our like beaver, not Northwest salamanders, but some of the like amphibian species. But in terms of the indicator species for the metric that we're planning, we don't have those picked out yet. So there are certainly lists in terms of, the rich history of traditional methods that are out there. And part of the kind of big question that's going to come out that we're hoping to answer from these paired kind of traditional sampling methods versus eDNA is to have an idea of, well, which of those species do we pick up on eDNA and which are maybe different, but they're unique DNA signatures that we find. So there was some work done in the EU, specifically creating algal-based metrics. And one of the things that they found, they were comparing kind of the morphological, which is kind of the like, you collect something, you use a microscope and ID it, and then the like DNA-based. And they found that they didn't necessarily find one-to-one species, but they found similar like structures basically of species at similar sites. And so they could build something that was equivalent. It was different, but it was equivalent in terms of its ability to say like, this site has this kind of quality and this site has, a lower quality or something like that. So it's a little bit of an unknown, like, we know, but we also, it's a matter of what all we can detect. Yeah. Thanks for that question.
Were any of these studies initiated through your RFP process?
Mm-hmm. I think all, except for maybe the pilot. Was that an RFP? RFP? Yeah. So they've all been RFP projects. Yeah. Yeah.
Any other questions in the room or on video?
Do you get any strength of signal information, or is it purely presence and
absence? That's one area that I think eDNA is really trying to figure out. I'm sure if you've been to any eDNA talks, there's a lot of talk. In terms of the sequencing base, so what I talked about last, we're still really at abundance, or not abundance, at presence absence. You get read abundances, but there's so much that can go into biasing the amount of reads you get for a particular species that you can't say like, if we get 10 DNA sequences of this and 100 of this, that's necessarily representative of the community. In terms of the single species assays, we're getting closer, I would say, but there's so much that goes into, so I guess the short answer to your question is no, we're not there yet. I hope that we can get there. There are some really promising studies that have looked at characterizing things like flow, temperature, amount of light, turbidity, so it's kind of different characteristics of water, and then have looked at DNA concentration. And so there are some studies, I guess, that show that we can maybe in the future, but there are kind of confounding variables that are site specific that you almost have to have that kind of study yourself in those sites to be able to say like for that one. We do have an attempt as part of one of our RFPs to get some idea for our watershed. We have some samples on East Fork Dairy and on the Upper Tualatin River that were taken at the same time that the Watershed Council was doing their rapid bioassessments, where they take snorkel surveys and count juvenile fish. And so we're hoping that that will give us an estimate of at least summertime when we have juveniles in the water, right? That's kind of a baseline. I would love to go out and take like snorkel surveys and do like actual spawning counts, and then compare that to the DNA signal. Because one of the things that we have found, and that the literature also would suggest, but when salmon in particular spawn, they put a lot of DNA into the environment, right? And that gets picked up in the water. And so we found that those second peaks were like, oh, that's so big, and it's higher than when we see the fish actually passing. So this year we tried to, at the same time that we took some of those lower Tualatin samples, take samples of some of our tributaries. And we found kind of these, like it would be really, really hot or really high at one site, and then really high, the next month at a different site. And so we think we're seeing these changes in spawning behavior between streams, but we can't say that for certain. So yeah, that's my long-winded answer of a not yet, but hopefully at some point.
I forgot to mention that, to my knowledge, we're the only entity with an eDNA provision in their NPDES permit. So hats off to DEQ for being open to new technologies that are going to better help us serve the river.
Thank you. Invitation for public comment. We'll now welcome any members of the public who'd like to provide public comment. You'll have up to 2 minutes, and Katie will be our timekeeper. Neil?
Dale Fee. Just a couple of comments. Glenn Fee, Shrimp, Wallets, and River Keepers invited Scott, Summer, and Bob Gardner a couple of years ago to come to his site and talk about PFAS and where we are. So to me, this is a very enlightening scientific presentation. It really pleases me to know that Clean Water Services is on top of it and is spending the thousands of dollars to be one of the best scientific labs around. That really pleases me. Because Clean Water Services is science-based, and I wish the federal government would be currently science-based. And I'm giving this a book away on tyranny, 20 Lessons from Snyder. If you want one, it's what you can do, maybe calendars, some of the things that happened in Trump advance. That's a side thing. So science is important. Sources intel, I mentioned Ronald Acres is the number one thing, the big industrial thing. And so I'm really pleased that Bob Gardner's worked and done that and found out that Intel has had a higher incidence of PFAS coming from it. And that the semiconductor industry cannot produce the chips without PFAS chemicals. They just can't do it. So what they're trying to do is to decrease the amount that comes out, and then DEQ does a hazardous site for that disposal of that. So DEQ, I think over in the Columbia River, Arlington area, they have a hazardous site where they now deposit the PFAS waste. Okay, so I think I'm pleased with DEQ. I noticed that Dean is on, I don't know if Dean from Stoll Reeves is here. I'm curious to know my attorney from Intel, because Intel is represented by Stoll Reeves a lot. Is he still on? I'm curious to know why he's here. I'm just curious about that. So I guess that's about what I have wanted to say. I'm just pleased. 2 minutes is up. 2 minutes is up. It's time to quit.
Thank you. Okay, announcements and Elizabeth, announcements for this meeting or next meeting?
Sure, so I do have a couple of announcements. First, I want to thank everyone who completed the CWAC engagement survey. Really grateful for the time that you took to provide feedback, and I just want to share a few takeaways that I gleaned from that information and how we're using that going forward. So legislative updates are popular. You'd like those to continue. PFAS, Scoggins, climate, West Basin Master Plan, design and construction standards were all also areas of desired additional discussion, presentation, engagement. We heard call outs in some of the comments about getting more information on how we work with other local agencies when it comes to water quality in the 12 River Basin, and an overview of shade credits, restoration efforts with partnerships, and maybe talk about what we're planning or what CWS does in the UGB areas regarding planning, and specifically how that works with the housing crisis. So we're taking all of these sort of under consideration as we're plotting out future sessions. Overall, folks thought dinner was either just right or maybe a little long. Keep me posted if that changes. Cadence of the meetings was either just right or maybe a little too frequent. No one asked for more, so my feelings are hurt. Data. Largely prefer in-person, but appreciate the opportunity for hybrid. Majority of folks are satisfied, but there are some things that we can do better. And I just want to highlight a particular call out and desire to lean into the advocacy part and the advisory role of the Clean Water Services Advisory Commission. So while this evening was largely informational, and hopefully you saw some of the tie-ins to the permit, to what we're trying to do to get ahead of regulation and rulemaking and other laws that might be coming down the pike, I want to preview that we're going to be leaning more into your advisory role this sort of spring, summer, and fall. You'll hear updates on West Basin Master Plan and some draft recommendations for I know a subcommittee has been meeting. That process is moving forward, and so we'll get your engagement and feedback on that. And later in the year, we'll be coming to you to talk about design and construction standards. So some meaty work to be done where we're going to be looking to you all for engagement. So just hopefully you enjoyed tonight, but if tonight's not your thing, there will be more coming in different types of work where we're going to really lead on this group. I'm more grateful for your time and engagement. Also wanted to share, we had mentioned there was going to be an air curtain incinerator demonstration from the Emerald Ash Borer discussion. We'll circulate information, but it's going to happen on April 9th between 10:30 a.m. and 3 p.m. at 12 River Farm. So we'll send out more information and emails if you want to attend and see what that technology looks like, which is pretty cool. And last but not least, our next meeting will be online. That's the end of my announcements.
Is that here?
What was that?
Is that here?
I have to check. Would you like it to be here?
Sure.
The invite says here. Okay, then it's here. Thank you.
Thank you, Elizabeth. Okay, well, all the speakers, thank you for all the great information, keeping us on time on our agenda. Really appreciate that. With that, we'll adjourn the CWAC for tonight.
Thank you all. Good night.
One of these buttons is big enough.